Hi everyone, this is Lata Krishna from EDUHUNT IIT and NEET Foundation Academy. We have learned about various strategies how plants reproduce, how plants multiply. We have also seen natural methods, and now we entered into the details of the artificial methods through which we propagate plants. Why propagation? Because every organism tries to improve, tries to survive, tries to cope up with the environment, tries to survive, tries to fight with the various factors of the environment. During this process, they adapt themselves to the environment. Environment is a dynamic one. Ecosystem is not a static one, it is a dynamic one. So, the changes that occur in the ecosystem or in the environment adopted by the organisms, as per the changes, they modify themselves. Due course of time, they show some modifications and also they develop some sort of multiplication techniques. That is about natural propagation. While coming to the artificial means of reproduction or vegetative propagation, we follow a process known as cutting. And second is layering. Third propagation method that actually the nursery people follow is grafting. We have seen the first two techniques, that is cutting and layering. In this session, we are going to know the details of grafting and the process types. Let's move on. What is grafting? It is nothing but joining of two different plants. There is a basal plant part, or the plant which has roots and which is in contact with the soil. That part of the plant is known as the stalk. It is the rooted shoot of one plant is known as the stalk, and it is joined with a piece of another plant known as scion. In grafting, there are two parts of plants. They are taken from two different plants. రెండు రకాల మొక్కల నుంచి రెండు రకాల పార్ట్స్ తీసుకుంటారు. రెండు రకాల పార్ట్స్ in the sense two different stem cuttings. One is not a cutting, actually the apical portion, the upper portion is cut, it is separated. The basal part which has roots is known as stalk. It is one part of the grafting technique. And the second part is it is obtained from another plant. The stem cutting of another plant, it is taken and inserted into the stalk. This is known as scion. That is, in grafting, while we do this technique, we should have two parts from two different plants. The basal part is known as the rooted part is known as the stalk, and the second part is known as the scion. It is the stem part of another plant. The scion is inserted. How it is inserted? a small insertion is made on the stalk, and it is now the scion is cut in such a way that it should fit in the incision made in the stalk. So I'll show you the technique. Let's watch. Let's erase this. Yes. Now watch from the beginning. Yes. See carefully. Observe it. A basal stalk is taken. A cut is made in the stalk. Another portion, stem cutting from another plant is taken and inserted in the stalk. Now, see, I'll label these two. Yes, now see, from here this portion is removed to make an insertion in the stalk. So the basic part is known as the stalk. Okay? The part which has in connection with the soil is the stalk. An incision is made here and the part is removed. Next, slowly observe. Yeah, that nodal region is removed in this example. Now, see another. Yes, this is scion. A part of a piece of stem with nodal region is removed from another plant and it is inserted in the incision made in the stalk. Now, the joint is sealed with the help of a tape. This is a grafting technique. After 2 weeks, 2 weeks later, it gives out the new branch. How simple it is, isn't it? Yes, you can also perform the same technique at your home. Try it once. Number of times you do, you will learn something from this practical. Okay, let's move on to the types of grafting. How many types of grafting are there? There are basically tongue or whip grafting, and second is wedge grafting, and third type of grafting is crown grafting. There are 3 basic types of grafting. The first is the tongue or whip grafting. Second is the wedge grafting. Third is crown grafting. And besides these technique called bud grafting is also there, in which only a single bud along with a small portion of stem or a bark is inserted near the cambium. Like as we have seen in the video, we have seen that a person is placing the scion in the incision made on the stalk. In the same way, here bud is placed near the cambium and it is employed for propagation. Bud grafting is widely practiced in especially roses, plums and apples, where a small portion of bark having intact cambium inserted instead of a scion is placed. The main portion in bud grafting is the bud. It is preferred in roses and also plums and apples. Rose is an ornamental example and plums and apples are fruit trees. These are the grafting techniques. And yeah, in this slide you can see different types of grafting. It is the stalk actually. In the stalk, the scion or the bud acts as the scion here. See, the bud is placed, an incision is made up to the cambium and the bud is removed from another plant and is placed in the stalk. This is a bud grafting. And approach grafting is another one where to the intact plant the graft is the scion is connected. It is the tongue grafting. Tongue grafting is the incision is made in such a way that it looks like V. And in crown grafting, the stalk must be wider. They made number of incisions and we have to insert the scions in the incisions. So that is crown grafting. At the tip of the stalk, we insert the scions. That is called crown grafting. And in wedge grafting, see how it is inserted. Only single scion is inserted. Here, the incision is made like V in the like wedge. It is like V in the stalk and the scion also removed and it should be placed in the stalk. The V portion, the empty portion, that is the V gap. In this gap, the scion should be inserted like this. It is the scion. It is stalk. Like this, we do wedge grafting. In tongue grafting, it is like this. A scion has buds even. That portion is inserted on the stalk. And these are the various types of grafting techniques. Yes, this picture will give you a clear idea about the grafting. This is a scion, this is a stalk, and during grafting an incision is made and inserted. The scion is inserted and the joint is wrapped with the help of a tape or a thread. So this is how grafting is performed. I hope you all have understood the grafting technique and types of grafting. Let's move on to the next one, that is micropropagation, which is another mode of artificial reproduction, artificial vegetative propagation. Micropropagation, it is micropropagation is also known as tissue culture. Tissue culture. How tissue culture is performed? What is meant by micropropagation? Propagation is multiplication. Micro in the sense in a very limited space, that is in the test tube everything is performed. In the laboratory itself it is done. So micropropagation is nothing but tissue culture. We require seeds for plantation. if we sow a single seed in the soil, it takes water from the soil, water and minerals, and it absorbs energy from the sunlight and CO2 from the atmosphere, and it has chlorophyll in the cell. So it performs photosynthesis, it grows, and it gives rise to a new individual. It gives a new individual. This is how seed germinates and gives a new individual, develop into a new individual. In case of tissue culture, if we place a tissue in the soil, it cannot germinate, it will not give rise to a new plant. For that purpose, we need to create an artificial environment which facilitates the growth and development of the tissue. And plants have a special capability known as totipotency. 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 It is nothing but the inherent capability of a plant cell to develop into a complete plant. That is known as totipotency. Whereas animal cells do not have this power. The power of generating a complete or developing into a complete individual from a single cell is known as totipotency. The power, the plant cell has that power. If we keep a single plant cell, which is a live cell, in a nutrient medium, it will give rise to, it will develop, the cell divides and develop into a complete plant. This inherent potency is known as totipotency. The all micropropagation technique, all the or the tissue culture technique depends on this capability of the plant. Micropropagation is nothing but propagation in a small environment or in a test tube. This is called tissue culture. It is performed in the laboratory, and I'll show you the picture how the plants were obtained during tissue culture technique. Yes, these are test tubes. You can see clearly the test tubes. These are 3 test tubes in which plants were there, small plantlets are there. This is the nutrient medium. మనము ఒక సీడ్ని సాయిల్లో వేస్తే ఇట్ జర్మినేట్స్. ఎలా జర్మినేట్ అవుతుంది? ఇట్ అబ్జార్బ్స్ వాటర్ అండ్ మినరల్స్ ఫ్రమ్ ద సాయిల్ అండ్ స్మాల్ రాడికల్ గివ్స్ అవుట్. దెన్ it gives out a small stem and leaves. It germinates. And after this, how it is germinating, it requires energy. It takes energy from the sunlight. That means it performs photosynthesis. It takes CO2 from the atmosphere. In this case also, the cell requires energy. Cell could not perform photosynthesis alone. We need to supply the minerals and nutrients because it is not in the soil. If we place the cell in the soil, what happens? Immediately the microorganisms attack the cell and it may get damaged. Because of that, we take proper care and place the cell on a nutrient medium which is a sterilized one, and place this in a sterilized room. So all the experiments should be performed under aseptic conditions. There should not be any microorganisms. Like ICU ఉంటుంది కదా, ఐసీయూలో ఉన్నప్పుడు చూడండి ఎంత కేర్ఫుల్గా ఉంటారు, ఎలాంటి ఇన్ఫెక్షన్స్ రాకుండా ఎంత కేర్ తీసుకుంటారో మనం కూడా ఇక్కడ అంత ఇంటెన్సివ్ కేర్ తీసుకోవాలి. మైక్రో ప్రాపగేషన్ ఆర్ టిష్యూ కల్చర్ చేసేటప్పుడు ఒక స్మాల్ సెల్ని లేదా ఒక చిన్న పార్ట్ ఆఫ్ ప్లాంట్ని స్టెరిలైజ్ చేసి న్యూట్రియంట్ మీడియం. వాట్ ఈస్ న్యూట్రియంట్ మీడియం? న్యూట్రియంట్ మీడియం ఈజ్ నథింగ్ బట్ ఇట్ హ్యాస్ సమ్ న్యూట్రియంట్స్ ఇన్ ఇట్. న్యూట్రియంట్స్ ఉంటాయి ఇందులో ఇన్ ద ఫార్మ్ ఆఫ్ సాల్ట్స్. వి డిజాల్వ్ సాల్ట్స్ ఇన్ వాటర్ అండ్ వి ప్లేస్ దట్ cell or tissue in this nutrient medium. Then after some time the cell undergoes cell division, give rise to a small mass of cells known as callus. A small mass of cells. Undifferentiated mass of cells is known as callus, and after some time, after few weeks, the callus, with the help of hormones, phytohormones like auxins and auxins and cytokinins, it organogenesis occurs and it forms roots and stem. Small plantlets will arise from the callus. This is known as organogenesis. Organs, that is root, stem, leaves, all these things emerge from the callus. This is known as, in the small space, the cell is giving rise to number of plants. Number of plants, that is multiplication, propagation. Under aseptic conditions, we are doing this. This is known as micropropagation. It is micropropagation. ఇవన్నీ ఎందుకు ఎసెప్టిక్ కండిషన్లోనే చెయ్యాలి అంటే, ఎసెప్టిక్ కండిషన్స్లో అంటే స్టెరిలైజ్డ్ రూమ్లో, స్టెరిలైజ్డ్ కండిషన్స్లో చెయ్యకపోతే మైక్రో ఆర్గానిజమ్స్ వచ్చి అటాక్ చేసి స్పాయిల్ చేసేస్తాయి సెల్ని. ఎందుకంటే న్యూట్రియంట్స్ మనం ఇచ్చాం కదా, న్యూట్రియంట్స్ ఎక్కడుంటే అక్కడికి వచ్చి మైక్రో ఆర్గానిజమ్స్ అటాక్ చేసేస్తాయి. So this is the end of the class, my dear children. I hope you all have understood about the various artificial methods of plant propagation. One is the cutting, second is layering, third is grafting, fourth one is a very advanced technique and it only performed by the scientist, not by a farmer. All the three techniques, that is layering, grafting and cutting, will be done by the farmers and even a nursery people can do. But the micro propagation, that is what we have learnt recently, is done only by the scientists. I hope you have understood the topic, and once more you listen to my videos, you will get complete knowledge on the subject. Thank you.